East of New Zealand, the Kermadec Trench begins as a shadow beneath restless South Pacific water. Its floor descends into the Hadal zone beside a chain of volcanic islands and submerged peaks. The wildness here comes from convergence, steep terrain, earthquakes, eruptions, and life under crushing pressure. Each force leaves evidence, but none can be understood by staring at depth alone. From the surface, you cross the boundary without seeing the descending plate beneath the ship. The mystery is how one narrow corridor can connect planetary recycling with specialized deep-sea ecosystems. Its walls are not smooth, and its axis does not remain equally deep along its length. Faults interrupt slopes, canyons guide sediment, and ridges divide neighboring pockets of habitat. Mapping reveals a dramatic form while raising questions about how quickly that form changes. Some changes unfold over millions of years, while earthquakes can rearrange slopes within minutes. Particles from sunlit waters descend through kilometers before reaching this permanently dark landscape. Their arrival connects surface seasons with animals that never experience daylight. The investigation enters through that descending material. Then follows geology, evidence, and adaptation together. Every observation occupies a tiny fraction of a trench stretching across a vast oceanic boundary. Wildness, in scientific terms, means interacting extremes whose details remain incompletely sampled. The Pacific Plate subducts westward beneath the Australian Plate, along the Kermadec margin. Northward, this boundary continues toward Tonga, while southward it approaches New Zealand's complex plate junctions. The descending oceanic plate is old, cold, and dense compared with the mantle beneath it. As it bends, faults open across its upper surface and allow seawater to penetrate fractured rock. Water carried downward influences, mineral reactions, and later contributes to melting above the slab. Magma rises through the overriding plate, feeding volcanoes of the Kermadec Island arc. That relationship links trench depth with volcanic activity, although eruptions do not originate inside the trench. Behind the arc, extension in the Havre Trough shows that the overriding region also deforms. The boundary, therefore, combines compression at the trench with stretching farther west. Multibeam sonar maps depth by recording echoes returned from overlapping strips of seafloor. Seismic networks locate earthquakes and estimate how faults moved during rupture. Deep-sea landers carry cameras, baited traps, and sensors into places ships cannot continuously observe. Sediment cores recover layered material that can preserve gradual settling and sudden gravity-driven deposits. These four methods provide complementary evidence while introducing different sampling biases. Sonar resolves landforms, but cannot identify every process that created them. Earthquakes reveal active fracture, yet silence does not prove a fault is inactive. Landers show behavior within a limited illuminated area and may attract scavengers toward bait. Cores preserve sequences, although erosion can remove layers and mixing can blur boundaries. The trench exceeds 6 kilometers across much of its deepest corridor. At such depths, pressure changes protein function, membrane structure, and cellular chemistry. Temperature remains near freezing, and food generally arrives from production far above. Oxygenated water still reaches the bottom, preventing depth from becoming a universally stagnant realm. This verified setting frames every biological interpretation made from Kermadec samples. The first sharp question concerns why some animals appear at particular depth bands. Hadal snailfishes have flexible bodies, reduced bone mineralization, and biochemical adaptations associated with high pressure. They detect prey and carrion, approach feeding opportunities, and use suction to capture small crustaceans. This Q-action sequence turns sparse food patches into growth, but it also exposes them to baited sampling. Amphipods respond rapidly to chemical traces, swim toward carrion, and consume tissue before competitors disperse it. Their speed helps recycle episodic food, falls through a community otherwise limited by downward carbon flux. Sea cucumbers cross soft sediment, ingest surface deposits, extract organic material, and leave process trails. That behavior mixes the upper seabed and changes microbial access to the buried particles. When you watch a lander recording, the illuminated animals represent attraction, endurance, and chance together. Counts near bait cannot be translated directly into natural density without additional controls. Pressure also sets physiological boundaries that differ among lineages rather than imposing one universal limit. Species replacement with depth may reflect pressure, temperature, food, history, and barriers between trench sections. Genetic comparisons can test connectivity, but small samples may miss rare or widely dispersed populations. Physical isolation matters because trench ridges and currents can restrict movement along the axis. However, similar environments do not guarantee identical communities, and different environments can host related species. The evidence supports ecological filtering without reducing every distribution to a single cause. Geology sharpens the problem by repeatedly disturbing the habitat being measured. Earthquakes can destabilize slopes, sending sediment-laden flows down canyons toward deeper basins. Fresh deposits may bury organisms, deliver organic matter, and create new surfaces for colonization. Pores sometimes show coarse or rapidly emplaced layers consistent with such events. Assigning a layer to one known earthquake requires secure age control and regional correlation. Without those tests, a disturbed bed records energetic transport, but not a proven historical trigger. Volcanic ash can also reach marine sediments, providing material and sometimes useful chronological markers. Currents may rework ash, so its final position need not mark simple vertical settling. The trench floor is therefore an archive, with missing pages, rearranged paragraphs, and uneven preservation. Interpreting it requires testing several transport mechanisms against grain size, chemistry, and geometry. This evolving landscape matters to life because disturbance resets habitats at different scales. Microbes colonize newly deposited sediment, metabolize accessible compounds, and alter surrounding pore water chemistry. Burrowing and feeding animals then redistribute particles, progressively softening the signature of the original event. Where organic matter accumulates, respiration can increase, even though bottom water remains oxygenated. Where slopes shed sediment, exposed surfaces may contain older material and fewer fresh food particles. The topography thus channels both physical energy and biological opportunity. If you descended through the Kermadec water column, light would vanish long before the trench appeared. A submersible's lamps would isolate fragments of slope, making navigation dependent on acoustic mapping. Pressure would test windows, seals, batteries, and every connection exposed to seawater. Robotic vehicles avoid human risk, but still face limited power, communication delays, and difficult recovery. These constraints explain why absence from footage is weak evidence of biological absence. They also explain why repeated transects matter more than one spectacular encounter. The Kermadec region supports broader questions about how Hadal species disperse between separate trenches. Larvae, adults, currents, and geological history may each contribute depending on the organism. Population genetics can distinguish recent exchange from long isolation only when sampling is geographically adequate. Conservation questions arise because deep ecosystems recover on timescales that remain poorly measured. Pollutants transported through ocean circulation and sinking particles can reach depths far from their sources. Detecting contamination does not automatically reveal biological harm, which requires exposure and effect measurements. The trench, therefore, connects distant human activity with environments once assumed completely remote. Its scientific value lies in repeated natural experiments across depth, disturbance, and isolation. Future exploration must sample those gradients without confusing technological reach with complete knowledge. Higher resolution repeat mapping could identify slope failures and newly deposited channels after major earthquakes. Long-term seismometers could locate small ruptures that temporary surveys miss. Unbaited imaging, paired with baited landers, could separate background movement from attraction-driven behavior. Matched sediment cores from slopes and basins could test how topography controls event preservation. Environmental DNA may reveal biological presence, but currents and degradation complicate location and abundance. Specimens remain necessary for anatomy, physiology, taxonomy, and verification of genetic signals. Pressure-retaining samplers could examine living responses without the abrupt decompression caused by ordinary recovery. Such experiments must distinguish adaptation from short-term stress during capture and handling. Broader seasonal coverage could connect surface productivity pulses with delayed food delivery to Hadal animals. The strongest future evidence will combine repeated measurements across the same precisely mapped locations. Even then, rare earthquakes and slow ecological recovery may outlast individual research programs. Uncertainty will narrow unevenly, with some processes remaining accessible only through indirect records. The opening shadow east of New Zealand will never become fully visible at once. Yet each mapped fault, dated deposit, and documented feeding sequence makes its interactions clearer. Kermadec remains wild not because evidence fails, but because active geology and Hadal life keep changing together.